This is a tutorial for your 5.16 Fixation and Root Nodules lab. So we're going to go over the lesson, what you're supposed to do, and then we'll go over your lab reports, and I'll help you answer some of the questions correctly. So always make sure that you read your lesson introduction. It gives you some important information on what you're going to be doing. So this lab is on nitrogen-fixing bacteria. And these bacteria form a symbiotic relationship with some plants, but not with others. So remember that symbiotic means that the relationship is helpful for both. So it's helpful for the bacteria and for the plant that they are living on. So nitrogen-fixing species of bacteria are either free-living in the soil, or they can inhabit cells of the organisms that benefit from their nitrogen-changing ability. Bean plants and other are members of the legume family of plants form symbiotic relationships with these type of bacteria. And we know that these relationships are present because there are root nodules. There's these little like nodes on the roots that you'll notice, and that's the bacteria. Okay, so you're going to gather your materials. So your student guide and lab instructions, make sure to read over this. Okay, so these are some optional resources I put in there. Let me just go back to the main menu and I'll show them to you. We don't have to go through them right now. These are just to help you with different things that you might encounter in the lab, like the microscope, so using a microscope correctly. There is another Explore section on the nitrogen cycle, and this will help you to get a better picture of what's going on in this lab. Okay, so we're going to click on Laboratory. Okay, so remember, a symbiotic relationship is a beneficial relationship. So two organisms are living together in a mutually beneficial way. Okay, so we were talking about a type of nitrogen-fixing bacteria of the genus Rhizobium. So in your student guide, you can take notes. So that's why I suggest opening it up and going through it as you go through each of these slides. Also, if you click on the definition, or the hyperlink, you can get a definition. You can also get
symbiosis.
If you don't know how to pronounce a word, it will pronounce it for you. There's a lot of difficult science vocabulary that are difficult to pronounce, so that's super helpful. Okay, so the bacteria live within and among the root cells of the plant, triggering the growth of structures called nodules. There, the bacteria convert nitrogen gas into usable forms of nitrogen for the plant, especially ammonia, which is NH3. This process is called nitrogen fixation. Okay. In return, the plant provides the rhizobium with a safe place to live, as well as carbohydrates and other organic compounds that the bacteria need for life. So they're both doing something for the other. So it's mutually beneficial. So you can see in this picture, these are legume roots, so bean plants, and they have little nodules on the roots. And this is where the rhizobium are actively fixing nitrogen for the plants. Okay, so these rhizobium don't form a relationship with all plants. It's only certain ones. They tend to prefer legumes like bean plants. And the nodules on the roots grow in two phases. So there are green nodules, and this indicates that the bacteria are reproducing and not able to perform their nitrogen-fixing duties. When the bacteria are actively fixing nitrogen, the nodules have a pink color. So the pink is the color that benefits the plant. The rhizobium can't reproduce and fix nitrogen simultaneously. They can do only one or the other at any given time. So it's kind of like a cycle. Okay, so through a microscope, nodule cells with rhizobium are distinguishable from rhizobium-free cells. Initially, as the nodule forms, its cells are similar in size, but when the bacteria enter the cells and begin to reproduce, the cells get bigger or expand. This makes room for the growth of the bacteria, or the rhizobium colony, because there's more than one bacteria in there. It's a colony of bacteria. And so the cell becomes larger than their neighboring cells that do not have rhizobium. So you can see the nodule right here. It's expanding, it's getting bigger. You can click here, you can see a little bit of a more detailed picture. So you can see the cells with rhizobium are much larger. The ones without, or the blue part, are much smaller. All right, the next part is the investigation. Okay, so you want to make sure you download your lab report, and I have it right here. This is the document you're going to be using to put your answers on and turn in. So there are 2 days, or you can finish it in 1 day. It's not too difficult. You should be able to finish it in 1 day. So it's a virtual lab. You don't have to do anything at home. You are observing the radish root. You can click on this and it'll give you some information on how to controls on a desktop and controls on a touch screen. Okay, so you can pinch and zoom with 2 fingers if you're using your iPad, or use the magnifier buttons and the drag function if you are using a computer. So if I drag and look at the roots, you're kind of like looking over it. And you are sketching roots and nodules here on a separate piece of paper. So you're looking at a bean and radish plants, okay? This is a bean plant. So you're gonna notice, you can see the little nodules on the bean plants. Let me see if I can move up. You can zoom in. So this part has no nodules, so there's no nitrogen fixing going on here, but here is where the bacteria are living and making the cells expand and get larger. Okay, so now you're gonna observe the nitrogen fixing bacteria in a legume root nodule. So you can see here, here's the root nodule of a legume. It's much bigger. And then you see these tiny cells over here. There's no bacteria there, so the cells are much smaller. Okay, so you're observing 3 pictures, and then you're going to draw them. So what you can do is, let's take a look. So here's question 1. Record your observations of the roots of the bean and radish plants in the appropriate data chart. Be sure to include a sketch of each plant's roots. So right here, we have the bean plant, okay? So they just inserted their own drawing here into my answer key. So what you could do is either you could use the draw function in Microsoft. So, okay. For the draw function, you can, I don't care what color you use, you can draw your own little roots. Let's get rid of the picture. Okay, draw. All right, so we have our roots there. You want to make sure that you have the nodules on there. Or your other option for drawing is that you draw it on a piece of paper, you take a picture of it, and you upload your screenshots into your answer document, okay? So to do that, you just click Insert. You can insert picture from file, and that will upload a picture there, okay? So your sketch for the bean plant should show a branching root form with lumps or nodules growing on the ends of the roots. The root branch at various angles, and it should look similar to the picture I showed you below. Okay? Okay, so the next one is the radish plant. That was the first picture we looked at. So you're going to do the same thing. You could use the draw function up here on Microsoft Word, or you can draw your own sketch on a piece of paper, like I said, and upload the screenshot. Okay, so are there nodules present? No. What are some similarities to the bean roots? So you could point out that plants have at least one root from which smaller roots branch out. So see, we kind of drew our main root down here. Let me actually add a little bit. You can have a main root here. It's like a little bit thicker, and then you have branching roots out from that, okay? Okay, so those are some similarities. So you can see that this has a main root as well, and then it has little tiny kind of shoots coming out. For the radish, it has one long main root from which tiny hair-like roots project. The bean roots are more uniform in size and branch out. So you want to give a description how they're different from the bean. And then you're going to sketch it. So your sketch should show a single long root from which tinier root hairs project out. And it should look similar to this picture right here. Okay, so part B. So this was picture B, and this is worth 10 points. You're going to sketch the bean nodule, including cells with and without rhizobium, showing what you observed in the micrographs. So here's a model answer. They have a picture of the root nodule. So you didn't need to, let me go back to the picture for you. And that was the last picture we looked at. So you didn't need to draw this over here. You need to draw this part right here, the part on the left, okay? All right, so once you draw it, you can label it. So this whole thing is the nodule. These are the cells without rhizobium on the outside because they're smaller, and the cells with rhizobium are on the inside. So here, right here, we have the cells with rhizobium. The cells in the blue portion are without. You have some more over here. Okay. Question 3. 2 new species of plants are introduced into an area that was low in available nitrogen compounds in the soil. One formed nodules and one did not. Which species would you reasonably expect to grow more robustly? So you would expect the ones to grow more robustly would be the ones with nitrogen-fixing bacteria because those help the plants to grow better. The plant's nodule formation indicates that there is a symbiotic relationship with the nitrogen-fixing bacteria, and that would cause the plant to have a greater benefit than if it didn't have the bacteria. So, because of the bacteria, the plant is receiving a constant source of nitrogen to meet its needs. So thus it's going to continuously grow better in its environment and probably be more abundant, okay? So you need to write your answer like that with complete sentences. No one-word answers, please. Okay, so our last question is question 4. How does the location of the nodules relate to the function of the nodule? So nodules, remember, are located in the roots, and the bacteria residing in the nodules are protected from the above ground environment. So the soil kind of provides some protection. There's a lot of nitrogen gas in the soil, which is good for the bacteria because they take nitrogen gas and they produce ammonia for the plants. So this constant supply of ammonia for the plants helps it to grow better. And so its need for nitrogen, and so does the number of nodules. So the greater number of nodules, the greater amount of nitrogen that can be produced for the plants. Okay, you're not copying this answer word for word. You are going to put it into your own words. This is called paraphrasing. If you copy word for word, you will receive a 0. All right, I'm sorry, so there was one more question. So we have cells without rhizobium and cells with rhizobium. You're going to label which is which. So I did that for you. Remember, the bigger ones have rhizobium in them and the smaller ones do not. You're going to then explain the differences. Excuse me. So remember that these cells with rhizobium are so much bigger because the bacteria is growing inside these nodules, causing the cell to expand and become bigger. The cells without rhizobium are a normal size. They don't have a need to expand. There's nothing growing inside them, so then they are smaller than the ones with the bacteria inside. Okay, so please make sure that you save your lab report. You come over to 5.17 assessment for this lab, and you will upload your answer document and submit it, and I will be happy to grade it. If you have any questions about this lab, please contact me. You can reach me through email or text message, and I'll be happy to help in any way I can. Thank you so much.